Alright, so in this lecture we're going to be covering energy and enzymes. So we have two different major forms of energy that can exist in a chemical reaction. That is your kinetic energy, which is the energy of motion, and potential energy, energy that is stored. So kinetic energy involves motion. Whenever we have motion, we're going to also have thermal energy, because when things are in motion, we're going to generate heat. Potential energy, major form would be our chemical energy. That would be the energy that is, once again, stored in chemical bonds. Remember that bonds are made up of electrons. Electrons like to constantly move. These electrons have a lot of energy. So two major forms of energy, kinetic, which is energy of motion, and potential energy, energy that is stored, energy that you have the potential to do something. So chemical reactions are going to involve energy transformations. We can never create new energy. Energy will only be transformed. So the amount of chemical energy in a bond is going to be based on the position of the electron and how far it is away from that positive nucleus. Once again, why is the nucleus positive? It is due to protons. Potential energy is going to depend on the position of those shared electrons. Weak bonds that have equally shared electrons will have a high potential energy. So a weak bond, high potential energy. Strong bonds have unequally shared electrons. Therefore, they will have low potential energy. So weak bonds, like our single bonds, are going to have high potential energy. Strong bonds, right, our double and our triple, are going to have a low potential energy. When we have a chemical reaction, right, when we're going from reactants to products, if the product formed has a shorter and stronger bond than the reactants, then the potential energy in the bond is going to decrease. So in this situation, if we have reactants going from products and the products have shorter and stronger bonds, potential energy in the bonds is going to decrease. Different energy transformations. Energy is going to be conserved. It can never be created nor destroyed. It can only be transferred and transformed. When we say transferred or transformed, that means, are we going from kinetic to potential? Do we have thermal energy? Things along that nature. Now we're going to discuss the concept of enthalpy. Enthalpy is the total heat content of a system. So enthalpy is going to include the potential energy of the molecule. Once again, potential energy is going to be related to the heat content. That's why enthalpy is basically your total heat content of a system. Your enthalpy will also include the effect of the molecule on the surrounding pressure and volume of that system. A change in enthalpy is going to be based on the differences in potential energy. So enthalpy, I want you to think about potential energy. Potential energy is going to impact the heat content of the system. Your delta H, or your delta change in enthalpy, is going to be therefore be based on a change in potential energy. Alright, different kind of reactions we have are exothermic and endothermic reactions. Exothermic reaction is when we release heat. We release heat when the enthalpy is less than 0. So in a change in potential energy, right, that change in heat content that a system has, when that is less than 0, it will be an exothermic reaction. That means we're releasing heat to the environment. Endothermic reaction is when our change in potential energy is greater than 0. Endothermic reactions are going to absorb heat from the environment. An endothermic reaction will take place when your products have a higher potential energy than the reactants. This is going to cause that positive enthalpy change, therefore greater than 0, therefore absorbing heat from the environment. Aside from enthalpy, we also have entropy. Entropy is the randomness of that reaction, right? How random are these molecules going to move in relation to one another? Therefore, we refer to entropy as the amount of disorder in a system. When the products of your chemical reaction become less ordered than reactants, your entropy is going to increase. So your delta S, right, your delta change in entropy, when that is greater than 0, your entropy is increasing because that system became less ordered. We went from a reactant that was highly ordered to a product that is less ordered, right? Has a lot of disorder. It's very all over. All these molecules are bouncing off one another. Your second law of thermodynamics explains entropy in a way that entropy is always going to increase in a system. We like to be disordered. We're always going to increase that entropy. How are we going to do that? Your products have to be less ordered than your reactants. Now your Gibbs free energy. Your Gibbs free energy is going to tell you whether or not that reaction is spontaneous or non-spontaneous. If the reaction is spontaneous, we do not have to supply any excess energy. The reaction will take place by itself. Formation of your phospholipid bilayer, that's a spontaneous reaction. Why? Your phosphate negative charge head will interact with the hydrophilic water. The lipid tail will interact with one another through hydrophobic interactions. That's a spontaneous reaction. How do we determine if a reaction is going to be spontaneous or not? We're going to look at that delta G. If the delta G is negative, it's spontaneous. If it's positive, it's non-spontaneous. So here's your standard free energy equation. Delta G is equal to delta H minus temperature in Kelvin times delta S. Your delta H, once again, is your change in enthalpy that represents your heat content and is dependent on potential energy. Delta S is your entropy, and our second law of thermodynamics states that we always want a highly disordered system. And T is going to represent your temperature in degrees Kelvin. When your delta G is less than 0, so when it's negative, typically, you're going to have a spontaneous reaction. Spontaneous reactions are going to be exergonic. When your delta G is greater than 0, you're going to have a non-spontaneous reaction. Non-spontaneous reactions are going to require an input of energy. Therefore, we call them endergonic reactions. So exergonic, release energy. Endergonic requires energy in order to proceed. When your delta G is equal to 0, your reaction will be at equilibrium. So let's talk about temperature and how concentration can impact the rate of a reaction. So in order for a reaction to take place, you're going to have to break bonds, right? Break the existing bonds as well as make new bonds. In order to break and form new bonds, your substances have to collide in a very specific orientation. Why? We have to make sure that these electrons involved in the bond formation or bond breaking are close to each other. If we have two electrons from two different molecules that are too far apart, there's no way we could form a bond. Therefore, in order to form these new bonds, we have to make sure that these atoms are close enough for the electrons to interact with one another. What will also impact the rate of the reaction is your concentration of your substrates, as well as your temperature. The higher the temperature and the higher concentration of these molecules you have, the more likely that they're going to collide with one another. Why? Your temperature is going to increase the speed of the reaction. Your molecules are going to get very excited and they're going to want to move all over. Right? Think about when you boil water. Think about those bubbles, right? That's your molecules moving very fast, very rapid, bouncing off of one another. Why? It's an effect of temperature. Higher concentration. The more substance you have, the more likely that these molecules will interact with one another and collide in that specific orientation. So your rate of the reaction is therefore going to be impacted by your temperature as well as concentration. Why? It's going to impact how these molecules collide with one another. All right, energetic coupling. We have our exergonic and endergonic reactions. Your exergonic reactions release energy. So in an exergonic reaction, you're going from reactants that have a high energy that turn into a low energy product. Because we're going from a high energy reactant to a low energy product, we're releasing energy. That released energy can then go on to drive an endergonic reaction, because endergonic reactions require energy. Endergonic reaction is going from a low energy reactant to a high energy product. So energetic coupling is explaining how exergonic reactions are going to supply the energy needed to drive an endergonic reaction. All right, our redox reactions, or reduction-oxidation reactions. Your redox reactions is going to involve the transfer of electrons. Oxidation is when we lose electrons. It's an exergonic reaction. Reduction is endergonic. Reduction is when you gain electrons. Oxidation and reduction always occur together. So as one substance loses electrons, another substance will have to gain those electrons. That's why they take place together. So when we lose electrons, your electrons are going to get transferred from an electron donor to an electron acceptor. Your electron donor is getting rid of electrons. It's losing electrons. So your electron donor will get oxidized. Your electron acceptor will grab onto these electrons and therefore become reduced. So an electron donor loses electrons, becomes oxidized, will donate those electrons to your electron acceptor. Your electron acceptor gains electrons and therefore is reduced. Your electron acceptors are gaining electrons, becoming reduced, and gaining potential energy. Every time we transfer an electron, we're also transferring a proton. So reduction, right, when we gain electrons, we're going to add protons. Oxidation, when we lose electrons, we're removing protons. I'm just going to add in a quick little mnemonic for you guys to remember oxidation and reduction reactions. So we always say Leo, the tiger, goes grr. Now, what does this mean? We lose electrons in oxidation and we gain electrons in reduction. Now remember that oxidation and reduction are both half reactions. They complete one another. They always occur together. Whenever we gain electrons, right, in reduction, we're also going to gain a hydrogen, a proton, and gain potential energy. Another way we can understand redox reactions is if we talk about specific electron acceptors. One of them that we have is flavin adenine dinucleotide. As you can see, your nucleotide bases, your nitrogenous bases are going to pop up over and over again. So your flavin adenine dinucleotide, or your FAD, is an electron acceptor that accepts 2 electrons. So it's gaining potential energy. It got reduced. Every time we gain electrons, we also have to gain protons. That's why we form FADH2. Another electron acceptor is nicotinamide adenine dinucleotide, or NAD+. Once again, it's going to accept 2 electrons. And also 1 proton to form NADH. Our FADH2 and NADH are going to be our electron carriers. Why are they electron carriers? They're readily accepting and readily donating electrons to other molecules. All right, here's just a little picture. Our flavin adenine dinucleotide, or FAD, we have 2 protons. FAD is going to serve as our electron carrier. It's going to accept 2 electrons, right? You see over here, it's going to accept 2 electrons in addition with 2 protons. When it accepts 2 electrons, your FAD is going to get reduced and increase potential energy and form your FADH2. Alright. ATP, our favorite substance in a cell, right? So ATP stands for adenosine triphosphate. ATP is your major energy source for the cell. We can't do anything without ATP. Now, why is ATP so good for energy transfer? It has to deal with our high energy phosphate bonds. So all these phosphate groups have a lot of negative charges, right? You see this negative oxygen, another negative oxygen, another negative oxygen. All these negative charges repel one another. Because these negative charges on the phosphate groups are repelling one another, they're going to have a lot of high energy bonds. So all these bonds are very high energy. High energy bonds have a lot of potential energy. How ATP works is we're going to start cleaving off phosphate groups. When we break these bonds is when we release that potential energy, and that potential energy will perform the work. Alright, this will explain this better. So ATP hydrolysis, right? Hydrolysis is when we cut with water, is going to release that free energy. Remember, ATP is a very high energy molecule. Why is it a high energy molecule? It has a lot of negative groups that repel one another. So hydrolysis of the bond between the 2 outermost phosphate groups, right? We see we have 3 phosphate groups. Hydrolysis between the last 2, right? Cleave off that last phosphate, right? We got rid of that phosphate here. When we cleave off that 3rd phosphate group, we're going to form adenine diphosphate, ADP, and release an inorganic phosphate. This is a highly exergonic reaction. Exergonic meaning we're releasing energy. How is your ATP going to drive an endergonic reaction? So ATP is an exergonic reaction when we hydrolyze off that last phosphate. Now we have energy, right? We have that individual inorganic phosphate that's a very high energy molecule. So how is ATP now going to perform work? Your energy release during ATP hydrolysis, right, when we cut off that phosphate, is going to get transferred to a substrate by phosphorylation. Phosphorylation just means we're adding a phosphate group. When you add a phosphate group to another molecule, or any group for that matter, you're going to change the protein shape, which may make it more reactive or less reactive, depending on the molecule itself. Alright, so just to recap your ATP, because I know it can be a bit confusing, ATP is your major energy source of the cell. ATP stands for adenosine triphosphate. Triphosphate for 3 phosphate groups. ATP is a high energy molecule because your phosphate groups are negative. All these negative oxygens are unstable, right? They repel each other. Because they're all repelling each other, these bonds are very high energy, and high energy bonds have a lot of potential energy. So when we want to perform some sort of job in our cell, we're going to do a hydrolysis reaction of your ATP. Hydrolysis means we're going to cut with water, and what are we cutting off? That 3rd phosphate group. When we cut off that 3rd phosphate group, we're going to have an individual phosphate. That individual phosphate is highly exergonic, right? We're releasing about 7.3 kcals of energy at that time. That phosphate will now participate in substrate level phosphorylation, so that means we're going to add that phosphate to a specific protein. When that happens, we change the shape of the protein, which may activate or inactivate a specific reaction. Alright, enzymes. So we talked about our reactions, right? We understand exergonic, we understand endergonic. We understand how energy plays a role in this. However, all these reactions are too slow to take place. Biological reactions are only going to occur at a fast pace in the presence of an enzyme. Enzymes are proteins, and these enzymes are serving as catalysts. Catalysts speed up reactions. How does an enzyme work? They're going to bring different reactants together in a precise orientation so that they come close enough to interact with one another. By doing so, it's going to make the reaction more likely to occur, right? You can't make plans with someone who lives 5 million miles away, right? It's going to take too long for that to happen. But when that person come and visit you, and they're right in front of your face, then you can say, hey, yeah, let's go grab a cup of coffee. Why? That served as your catalyst. You're now in a precise orientation where you can interact. Another thing about enzymes is that they're specific for a single type of reaction. So an enzyme that speeds up one reaction, right, let's say it's going to speed up the breakdown of lipids, that enzyme cannot speed up a reaction that's going to break down sugars. It will never happen. So before a reaction can take place, right, before we can start mixing and mingling with our reactants first need to collide in a precise orientation, right? They have to be close enough. In addition, they have to have enough kinetic energy to overcome that repulsion between your electrons. Remember, electrons are negative. If you take two of the same charges, right, negative and negative, and try to bring them close to each other, they're going to repel. That's why that precise orientation is very important. Also, you need enough kinetic energy to overcome that. So our enzymes are going to bring substrates together. Substrates, right, are going to bind to an enzyme's active site. So an enzyme is going to have this little pocket called your active site. Your active site may have different amino acid tags or different sort of like sugar tags that a substrate will recognize and bind to. So your enzyme will bring substrates together. Your enzyme has an active site. Substrate will bind to the enzyme's active site. Bonds will break and form to generate products at that point. Many enzymes will undergo a conformational change in order to fit that substrate exactly, and we call that induced fit. So it used to be referred to as the lock and key model. Induced fit is more likely to happen. What induced fit means is, let's see this picture here. Okay, good. So here's your enzyme, the green. Here's your active site. Your active site's a little pocket that's going to be specific for a substrate. Your substrate will interact with the ATP in this scenario. Once they combine, they're going to have that special shape to fit into the active site. Then the reaction will take place in your active site and we will release a product. Induced fit means that this enzyme is going to conform around that substrate. Alright, so your substrates are going to bond through hydrogen bonding or other interactions with amino acid side chains at the active site. Once your substrate binds to the active site, whether it's through amino acid interactions, right, with our R groups, or through hydrogen bonding, we're going to form an unstable intermediate. That unstable intermediate is our transition state. In order to reach our transition state, we require activation energy. Your activation energy is required to strain the substrate's bonds so that it can reach the transition state. Right? Our transition state is going to be a very unstable intermediate. It's not happy. Therefore, in order to get to that unstable intermediate, we need a lot of activation energy. Reaction will take place when we have enough kinetic energy to reach transition state, and when the kinetic energy of molecules is a function of their temperature. Therefore, your reaction rates will depend on kinetic energy of reactants. Do we have enough kinetic energy to reach transition state? as well as the activation energy of the reaction. Alright, so here's your transition state. Your transition state, in order to reach that, we need high activation energy. Right? Our reactants have energy, and our products are going to have energy. This transition state is very unstable. It's not a happy intermediate. Right? So it requires a lot of energy to get to this point. Our enzymes are going to help by cutting that transition state activation energy in half. So interactions between the enzyme and the substrate are going to stabilize that transition state. Basically, what your enzyme is going to do here is going to make sure your transition state is more stable, right? We're going to decrease the workload it has to do. By doing so, we're lowering the activation energy required for the reaction to proceed. Another important thing about enzymes is that they can be reused. So you can use an enzyme over and over again without it being consumed by the reaction. Alright, how does enzyme take place? What happens here? So it has three major steps. First is initiation. Initiation takes place when our substrate is precisely oriented in order to reach our active site of the enzyme. Right? Initiation. We have to make sure we're close enough. Then we have transition state. Transition state's very unstable, that unstable intermediate. Your transition state is going to involve interactions between the substrate and active site R groups. Remember, our active site is very specific for a particular substrate. Why is it specific? It's going to have special R group amino acid chains to interact with the substrate. Then we have termination. Termination is when we release the reaction products from the enzyme, and now this enzyme can be used again. So three major steps in terms of an enzyme and a reaction. Initiation, we have to make sure it's close enough, right? We have to make sure that they can bind to that active site. Transition state, we're going to start changing our shape, right? We have to have that induced fit. We're going to have interactions with the R groups. This is very unstable. Then termination, you release the products and the enzyme will be used again. So what can impact the rate of an enzyme reaction, enzyme catalyzed reaction, is going to depend on your substrate concentration as well as your enzyme concentration. And this is going to be referred to as your saturation kinetics of your enzymes. When we have a low substrate concentration, the rate of the reaction is going to be higher, right? We have to convert this substrate into a product. In order to convert a substrate into a product, we need that enzyme, right? Because that's where the reaction is going to take place, at the enzyme's active site. If we only have to turn 2 substrates into products, we have an abundant amount of enzymes available, so the reaction will take place very quickly. Versus if we have 100 substrates to turn into products and only 10 enzymes. Your speed of reaction will therefore slow as the substrate concentration increases, right? If I have 100 substrates that I have to turn into 100 products and I have 10 enzymes, it's going to be a very slow reaction. It's going to slow down it. Eventually, we will reach a maximum speed at a high substrate concentration, right? Eventually, these enzymes are going to become overwhelmed. We can't increase their speed any longer. So all enzymes are going to show this type of saturation kinetics. Active sites cannot accept substrates any faster, right? We can't change the speed at which the enzyme is going to work. The only way we can change the speed of reaction is if we add more enzymes. So that's a good ratio between substrate and enzymes. Now your enzymes are special. They don't like to work alone. What does that mean? We're going to give them some cofactors, coenzymes, or additional prosthetic groups. So your cofactors are going to be like inorganic ions, right? Your zinc, your magnesium, and iron. They're going to reversibly interact with enzymes. Reversibly means they can attach onto the enzyme and then remove itself if it no longer has to work. Your coenzymes are organic molecules like your NADH and FADH2 that interact with enzymes, right? Your electron transport carriers. Then we also have prosthetic groups. Prosthetic groups are non-amino acid atoms or molecules that are going to get permanently attached to proteins. So cofactors are reversible. Prosthetic groups are irreversible. They're going to be permanently attached. Your cofactors, coenzymes, and prosthetic groups are important for enzyme function. What also impacts an enzyme is going to be its structure, right? Enzymes are proteins. Proteins are sensitive to environmental changes. Proteins are also dependent on proper folding. So your enzyme function is dependent on certain conditions. Temperature will impact the folding and movement of the enzymes and its substrate, right? Your temperature, high temperature will denature that amino acid structure. When we start to denature our protein, it's very hard to take that tertiary structure again. pH can also affect the enzyme's shape and structure. Each enzyme is going to function at an optimal temperature and optimal pH. The enzymes in your stomach that are involved with a lot of digestion, they have an optimal pH around 2. If we had, those enzymes will therefore not work at a pH of like 10. Majority of our enzymes are going to be regulated, right? We can't have reactions taking place all the time. We have to make sure that these enzymes are doing their job when they have to do their job and that they're not overstepping boundaries or being lazy. So regulatory molecules are going to control when and where an enzyme functions. These regulatory molecules can change an enzyme structure or its ability to bind a substrate. They may either activate or inactivate the enzyme's function. So a regulatory molecule is ultimately going to change the shape of the enzyme. If we change the shape of the enzyme, we can no longer bind to our substrate. If the substrate can't bind to the enzyme, we will not have a reaction. So, regulating enzymes through a non-covalent interaction. Non-covalent interactions will not permanently affect the enzyme structure. Why? They're reversible. Some of these interactions are going to include competitive inhibition and allosteric regulation. So competitive inhibition takes place when we have another substrate competing for that enzyme's active site. So competitive inhibition, other molecule competes with the substrate for the active site. If you have more substrates that serve as a competitive inhibitor, that higher concentration is going to overwhelm the enzyme, and then the enzyme will not bind its initial substrate. So the reaction will not take place. Allosteric regulation is going to be a molecule that binds to a site other than the active site on the enzyme. Allosteric regulators are going to bind to the allosteric site. This is still going to change the enzyme shape and can either activate or deactivate the enzyme. So competitive inhibitor will always deactivate. Allosteric will either activate or deactivate. So here's our competitive inhibition. Our red represents the substrate we want to turn into a product. The purple is your competitive inhibitor. It has a similar shape, similar R groups to your substrate. So it will bind to the active site and kick out our initial substrate. If this substrate cannot bind into the enzyme's active site, we won't get that transition site and we won't form products. So the reaction does not take place during competitive inhibition. Allosteric regulation is when we have another molecule that binds to the allosteric site of your enzyme. When we bind to the allosteric site of our enzyme, we're going to morph the enzyme shape so that in the case of regulatory activation, when our substrate binds, the enzyme shape changes and we're locking in these substrates. So the reaction will never stop. Allosteric inhibition, we bind the molecule to an allosteric site the enzyme itself changes, the active site changes. We can no longer fit the substrate into the active site. The reaction will not take place. Allosteric activation, when we're going to bind our substrate, then we bind an allosteric molecule. It's going to change the shape and then force these substrates to remain in the active site. So the reaction takes place over and over again. Regulation can also include covalent modifications. A covalent modification is going to change the enzyme's primary structure. Can be reversible or irreversible once again. If it's irreversible, that means that usually if resulted in cleavage of a peptide bond. Most common reversible modifications of enzymes is going to include the addition of a phosphate group, and we call that phosphorylation. The enzyme kinase is going to phosphorylate different enzymes. So kinase is responsible for phosphorylation. It's a common reversible modification, the addition of a phosphate group. All right, our metabolic pathways. Metabolic pathways is going to be a series of reactions. Each reaction is going to get catalyzed by a different enzyme, and that's going to build our biological molecules. There's two types of reactions, catabolic and anabolic. Catabolic is when we break down molecules. Anabolic is when we build molecules. So here is just a little representation of a metabolic pathway. We begin with reactant A. We get product B. Product B will then serve as a reactant to form product C. Each step is going to involve a different specific enzyme. We can also have feedback inhibition. So feedback inhibition is a way to regulate metabolic pathways. During feedback inhibition, you're going to have one of the pathways get inhibited based on one of the products. So if we have too much product, right? Let's say we just formed way too much monosaccharides, right? We broke down glucose. We broke down like the cellulose. We have too much glucose floating around. That end product of the pathway, so let's say they're glucose, will go back to the first enzyme, bind to that enzyme, and inhibit it. So if this enzyme can't work, we won't get this product. Then we won't get that product. And then we won't get our last step of just that single sugar. That would be feedback inhibition. Feedback inhibition, your end product is going to inhibit an enzyme. Alright, so this represents regular active enzyme. Right? We have low product, so we want to make product. Right? We're not happy. We need to make more product by converting the substrate, right, the reactant, into a product. Substrate binds the active site. We're going to get our intermediate. The intermediate binds the enzyme. We get another intermediate, goes to another enzyme, and we get our product. Let's say we form too much product, right? We no longer need to keep forming this product. The product will bind to the allosteric site of an enzyme. Now our substrate can no longer bind to the active site. So we won't activate enzyme 2 and therefore not activate enzyme 3. So feedback inhibition is going to shut off the entire pathway. Why? It's based on our end product binding to our allosteric site.